Hi everyone, welcome to the Physiology class on the Health Course channel. Today, we will start with the gastrointestinal physiology class. We are going to talk about motility and nerve control. The topics we are going to see in this class are some generalities: gastrointestinal physiological anatomy, gastrointestinal nervous control and gastrointestinal humoral control, and the types of movements in the gastrointestinal tract. The digestive system provides the body with a continuous supply of water, electrolytes, vitamins, and nutrients. To do this, our digestive system has to perform several functions such as the movement of food, the secretion of digestive juices and digestion, the absorption of nutrients, the circulation for the transport of substances, and the nervous and hormonal control of all these functions. Each function will be discussed in its respective class. In this class, we will discuss the basic principles of motility and nerve control. When we eat food, it enters through the mouth through the esophagus and meets the lower esophageal sphincter, and from there it passes through the stomach. The sphincters found in the stomach are the cardia in the upper part and the pylorus in the lower part. Then the food bolus goes through the duodenum, jejunum, ileum, where it meets the ileocecal valve that communicates the small intestine with the large intestine. Once it is in the large intestine, it goes through the ascending colon, descending transverse colon, and finally through the rectum. This entire journey is made from the mouth to the anus. The layers of the intestinal wall from the outside in are the serosa, the longitudinal muscular layer, the circular muscular layer, the submucosa, and the mucosa, which has a smooth muscle layer called muscularis mucosae. If we make a cross-section of an intestine to analyze its structure, we can visualize that it presents different layers. These layers are classified from the outside to the inside as the serosa, the longitudinal muscular layer, the circular muscular layer, the submucosa, the muscularis mucosae, which is part of the mucosa, and finally the mucosa. And the latter is where we can observe the intestinal villi. The circular and longitudinal muscle are connected to each other by a structure called gap junction. The function of the gap junction is to allow the passage of ions from one cell to another, which indicates that it functions as a true syncytium. Recall that the gastrointestinal muscle is of the unitary smooth muscle type, and when a stimulus arrives, it travels through all the muscle cells, thanks to the gap junction. This action takes place thanks to proteins called connexins. See, these are the connexins that are found between two cells. When an action potential arrives at a cell, all cells are depolarized together, and this is thanks to the connexins that allow the free passage of ions. Gastrointestinal smooth muscle is excited by slow, almost continuous intrinsic electrical activity that travels across the muscle membrane. This activity has two types of electrical waves: slow waves and spike potentials. These are the slow waves. The rhythm of almost all digestive contractions depends on the frequency of slow waves and the membrane potential of the muscle fiber. The slow waves are not action potentials, but are slow and undulating changes of the resting potential. These oscillate or vary between 5 and 15 millivolts. The frequency of slow waves varies from 3 to 12 waves per minute, depending on the location of the digestive tract. For example, in the gastric body we have approximately 3 slow waves per minute, in the terminal ileum from 8 to 9, and in the duodenum, approximately 12 slow waves per minute. It is believed that the origin of these slow waves is due to a complex of cells called interstitial cells of Cajal. The function of these cells is to function as electrical pacemakers of the smooth muscle. Just as the heart has its own pacemaker, so does the gastrointestinal tract, which are these interstitial cells of Cajal. These cells constantly send slow waves to the smooth muscle, and their function is to control the appearance of intermittent spike potentials. These are interstitial cells of Cajal where slow waves are generated by smooth muscle, and the passage of slow waves through smooth muscle. Remember, these slow waves do not induce muscle contraction, but control the spike potentials. The spike potentials are true action potentials, and it is these that will cause smooth muscle contraction. The difference between slow waves and spike potentials is practically that slow waves are not action potentials, whereas spike potentials are action potentials and will cause smooth muscle contraction. The main differences of the action potential of the nerve fibers and the gastrointestinal smooth muscle is that the gastrointestinal smooth muscle has between 10 to 40 times more duration than those of the nerve fiber and can be prolonged up to 20 milliseconds. Another difference is that, if you remember, for the depolarization of a nerve fiber, the rapid entry of sodium was required, while in the gastrointestinal smooth muscle the cell depolarizes thanks to the calcium-sodium entry. That is to say, the slow entry of calcium and sodium. In the gastrointestinal smooth muscle action potential, we have that in the resting state it is at minus 56 millivolts approximately, the excitation threshold minus 40. Here we have the slow calcium-sodium entry, and it is thanks to this slow calcium-sodium entry that makes the action potential last longer. There are factors that stimulate the muscle fiber, that is, they excite and depolarize it, such as muscular distension, acetylcholine, parasympathetic stimulation, which practically occurs through acetylcholine and other hormones. There are also factors that inhibit the action potential or hyperpolarize the cell, such as the stimulation of the sympathetic nervous system and its hormones, such as adrenaline and noradrenaline. Norepinephrine has the greatest inhibitory capacity because norepinephrine has a greater affinity for the alpha-adrenergic receptors. Adrenaline has an affinity for beta-adrenergic receptors, but alpha-adrenergic receptors are abundant in gastrointestinal smooth muscle. Because of this, noradrenaline has a greater effect on the gastrointestinal action potential, and an inhibition would obviously hyperpolarize it, thus leaving it more negative, and would practically paralyze the smooth muscle, i.e. there is no contraction and no depolarization. In smooth muscle contraction, the sarcoplasmic reticulum is a calcium store, and when an action potential arrives, it allows the entry of sodium and calcium was also released from the deposits. The calcium released would bind with calmodulin, forming the activated calcium-calmodulin complex, thus the enzyme myosin kinase. This enzyme would cause a phosphorylation of the myosin light chain, i.e. of the myosin head, causing smooth muscle contraction. Relaxation occurs thanks to another enzyme, myosin phosphatase, which causes the opposite, dephosphorylation of the light chain. Myosin kinase causes phosphorylation, whereas myosin phosphatase causes dephosphorylation of myosin light chains. We see here a membrane potential, a slow wave membrane potential, and the muscle tone in a normal state in a normal smooth muscle. When smooth muscle is stimulated, the spike potentials increase, and so do the slow wave frequencies and muscle tone. When it is inhibited by the factors that we saw previously as a sympathetic stimulus, we see that the membrane potential there are slow waves in smaller proportion, but notice that there is not even depolarization. There are no action potentials, there are no spike potentials, and the muscle tone practically does not exist since it is inhibited. The gastrointestinal tract has its own nervous system, called the enteric nervous system, which is located throughout the thickness of the walls from the esophagus to the anus, and is formed by two nerve plexuses, Auerbach's or myenteric plexus, and Meissner's or submucosal plexus. Auerbach's plexus mainly controls gastrointestinal movements, and Meissner's plexus controls secretion and local blood flow. The enteric nervous system can function on its own, but is influenced by the autonomic nervous system. For example, the sympathetic nervous system inhibits gastrointestinal functions, and the parasympathetic nervous system activates gastrointestinal functions. Auerbach's plexus is located between the longitudinal smooth muscle layer and the circular smooth muscle layer, while Meissner's plexus is located in the submucosa. Let's make a cut in the image, and here we can better appreciate Auerbach's plexus between the two muscle layers and Meissner's plexus in the submucosa. Auerbach's plexus is the most external and is located between the two muscular layers, and if we stimulate it, the following happens. We increase the tone of the wall, we increase the intensity of the rhythmic contractions, we accelerate the contractions, and we elevate the conduction velocity. Remember that the stimulation of Auerbach's plexus also inhibits two sphincters, the pyloric sphincter, which controls the emptying of the stomach, and the ileocecal valve sphincter, which controls the passage of food from the small intestine to the large intestine. Notice here are the pyloric sphincter and the ileocecal sphincter. Now Meissner's plexus is more internal and is located in the submucosa and acts controlling functions of segments of the internal wall, such as intestinal secretion, absorption, local contraction of the submucosal muscle. Here we can appreciate the layers of the intestine with the two plexuses, the layers that we saw previously increasing the myenteric nerve plexus or Auerbach's plexus, and Meissner's plexus or submucosal plexus here, which is located in the submucosa, and here we have the mesenteric, which is the one that covers the organ. In the control of the autonomic nervous system, we have the sympathetic and parasympathetic nervous system. The parasympathetic nervous system innervates the intestine through two divisions, the cranial division through the vagus nerve, and the sacral division through the sacral nerves. Its stimulus potentiates the enteric nervous system, thanks to its released neurotransmitter, which is acetylcholine. The sympathetic nervous system innervates the intestine by means of the sympathetic chains coming from thoracic segment 5 and lumbar segment 2, and form prevertebral ganglia here, as the celiac ganglion and the mesenteric ganglion. The sympathetic stimulus inhibits the enteric nervous system, thanks to its neurotransmitters, adrenaline and noradrenaline. But it is noradrenaline which inhibits practically all the gastrointestinal smooth muscle, except the muscularis mucosa, which practically does the opposite. It stimulates it, and a powerful stimulus can even paralyze all the gastrointestinal movement, that is to say, all the peristalsis. The parasympathetic releases acetylcholine and the sympathetic releases noradrenaline or norepinephrine, which is the same, inhibits the entire gastrointestinal system. Now a difference is that the parasympathetic has greater action on the portions closer to the mouth and closer to the anus, that is, its stimulus is more intense than the parasympathetic nervous system in these areas. Now the sympathetic stimulus has no preference and acts equally throughout the digestive tract. Here we see an image representing the intestine, Auerbach's or myenteric plexus and Meissner's plexus, and we see how the intestine receives innervation from the sympathetic nervous system. Practically all its neurons of the sympathetic system are postganglionic, and of the parasympathetic system are preganglionic, which we are going to see in detail in the autonomic nervous system class. And we have afferent sensitive nerve fibers, that is to say that they go from the digestive tract to the prevertebral ganglions to the spinal cord and to the brainstem. Many afferent sensory nerve fibers originate in the gastrointestinal tract and have their cell bodies in two sites, the enteric nervous system and in dorsal root ganglia of the medulla. These cell bodies can be stimulated by mucosal irritation, excessive distension of the intestine, by the presence of specific chemicals in the intestine. The substances transmitted by these fibers can cause either excitation or inhibition, depending on the type of reflex we are talking about. The main gastrointestinal reflexes in the enteric nervous system, the sympathetic nervous system and the parasympathetic system, maintain three main types of reflexes. These reflexes are completely integrated within the nervous system of the intestinal wall. Reflexes that go from the intestine to the prevertebral sympathetic ganglia and return to the gastrointestinal tract, and reflexes that go from the intestine to the spinal cord or brainstem and return to the gastrointestinal tract. The reflexes fully integrated within the nervous system of the intestinal wall already control mainly digestive secretion, peristalsis, mixing contractions and local inhibitory effects. Several of these reflexes we are going to see throughout the classes of gastrointestinal physiology. and the reflexes that go from the intestine to the prevertebral sympathetic ganglia and return to the digestive tract. We have the gastrocolic reflex, which are reflexes that come from the stomach and induce the evacuation of the colon, i.e. they come from the stomach and indicate the colon to evacuate. The enterogastric reflex, which are reflexes that come from the colon and intestine and inhibit these reflexes towards the stomach and inhibit gastric secretion. The colicoileal reflex is a reflex that comes from the colon and inhibits the emptying of the ileum into the colon, i.e. here of the ileocecal valve. Now we have the reflexes that go from the intestine to the spinal cord or brainstem and return to the gastrointestinal tract. Among them we have the reflexes originating in the stomach and duodenum that go to the brainstem and return through the vagus nerve or pneumogastric nerve to control the motor and secretory activity. We have painful reflexes that cause an inhibition and a general inhibition of the digestive system, and we must know that when there is pain, peristalsis is paralyzed. Finally, we have defecation reflexes, which are reflexes originating in the colon and rectum that go to the spinal cord and return to produce contractions in the colon and rectum itself, which are defecation reflexes. There are several reflexes, but these are the main ones that we will see in detail in their respective classes. The enteric system, like Auerbach and Meissner, is connected with the autonomic nervous system, sympathetic and parasympathetic, so they work together. The sympathetic system inhibits, and the parasympathetic system stimulates it. In the hormonal control of motility, the main digestive hormones are gastrin, cholecystokinin or CCK, secretin, gastric inhibitory peptide or GIP, and motilin. There are others, but we will see them in their respective classes, and these are the main ones. Gastrin, stimulus for the release of gastrin are proteins, distension and nerves, especially the pneumogastric nerve, and acid does the opposite, inhibits the release of gastrin. Gastrin is secreted in the G cells of the antrum of the duodenum and jejunum, and its action of gastrin is to stimulate the secretion of gastric acid and stimulate the growth of the gastric mucosa. We will see in more detail in the class on gastric secretion. The stimulus for the release of cholecystokinin or CCK is fatty proteins and acids. Cholecystokinin is secreted in the I cells of the duodenum and ileum, and its function is to stimulate pancreatic secretion of bicarbonate and enzymes and gallbladder contraction. It also stimulates the growth of the exocrine pancreas and inhibits gastric emptying. Cholecystokinin stimulates the secretion from the pancreas of bicarbonate and pancreatic enzymes, not to mention that it contracts the gallbladder and also inhibits gastric emptying. Now let's talk about secretin. The stimulus for secretin release is acids and fats. Secretin is secreted in the S cells of the duodenum and ileum. Its function is to stimulate the secretion of pancreatic bicarbonate pepsin and bile bicarbonate. The function of bile and pancreatic bicarbonate is practically to buffer gastric acids. It also stimulates the growth of the exocrine pancreas and inhibits the secretion of gastric acid. That is, apart from inhibiting the secretion of gastric acid, it also stimulates the release of alkaline substances, such as pancreatic bicarbonate and biliary bicarbonate, which have the function of neutralizing the pH of the food bolus in the intestine, coming from the stomach. Now let's talk about gastric inhibitory peptide. The stimulus for the release of gastric inhibitory peptide are proteins, fats, and carbohydrates. Gastric inhibitory peptide is secreted in the K cells of the duodenum and jejunum. Its function is to stimulate insulin release and inhibit gastric acid secretion. Now let's talk about motilin. The stimulus for motilin secretion are fats, acids, and nerves, especially the pneumogastric. Motilin is secreted by the M cells of the duodenum and jejunum. Its function, as its name indicates, is to stimulate gastric and intestinal motility. It stimulates both gastric and whole intestine motility. This motilin is secreted mainly in periods of fasting. Now, when we ingest food, the secretion of motilin is inhibited. In the digestive tract movements, the digestive tract has two types of movements: propulsive movements that move food at a speed suitable for digestion and absorption, and mixing movements that keep the intestinal contents mixed. Let's talk only about the generalities of these movements, since there will be an exclusive class called propulsion and mixing of food. When we talk about propulsive movements, we talk about peristalsis, which is the basic movement of the digestive tract. These peristaltic contractions occur thanks to the contraction of the smooth muscle and its gap function. The main stimulus for peristalsis is distension. When distended, the enteric nervous system creates practically a ring 2 centimeters above the distended zone. A contractile ring around this distended zone is called peristaltic contraction. Other stimuli besides distension are epithelial irritation, either physical or chemical, and obviously the parasympathetic stimulus mediated by acetylcholine. For an effective peristalsis, there must be an active Auerbach's plexus or myenteric plexus, since it is this plexus that generates the contractions. Let's talk about the law of the intestine. The peristalsis always goes from the mouth to the anus. We have to be clear about that. In case there is a depolarization of the smooth muscle towards the mouth, it would cause the stimulus that generates these contractions towards the mouth to stop. Now, when this demoralization is directed towards the anus instead of the mouth, this stimulation would continue for a much longer time, for a considerable time. Then the peristalsis of Auerbach's plexus goes in the anal direction, and does so by pushing the intestinal contents approximately 5 to 10 centimeters until this wave disappears. At the same time as the proximal part contracts, the distal part relaxes, which is called receptive relaxation. The sum of this peristaltic reflex and the peristaltic movement in the anal direction is known as the bowel law. There are two types of mixing: those produced by the peristaltic contractions themselves, and those produced by local constriction contractions. In some places, the peristalsis itself produces the mixing of food, for example, when the intestinal advance is interrupted by a sphincter. As we see in the image, notice that the gastrointestinal movement itself is interrupted by the sphincter, and mixing occurs here. Peristalsis produces the mixing against a sphincter. This peristaltic wave kneads the content against the sphincter and mixes it. There are also local constriction contractions, which are contractions that last between 5 to 10 seconds in one part of the intestine, and then every few centimeters. There are other local constriction contractions. The purpose of this mixing movement is to chop and crumble the intestinal contents, i.e. the food, and unlike the peristaltic waves, these contractions are local. As we see in the image, there are local contractions, but there is no movement resulting from the alimentary bolus. There is no forward or backward movement. They are simply local contractions. We will see the movements of propulsion and mixing of each digestive part in another class. As bibliography, I use the Guyton Hall Physiology Treatise, edition number 13.